have you, why don't I teach you about protein synthesis? Hi, my name's Summer, and I'm here with my partner Jody to tell you all about the processes of transcription and translation. First, let's tell the differences between RNA and DNA, the two micromolecules which contain the genetic code for all life. DNA, otherwise known as Deoxyribonucleic acid, is the hereditary material in humans and all other living organisms. For the eukaryotic cell, DNA is found within a cell's nucleus. The genetic information is contained within the sequence of the 4 nitrogen bases:
adenine, thymine, guanine, and cytosine.
Adenine pairs with thymine, and guanine compares with cytosine, forming units called base pairs. Each base pair attaches to a deoribose sugar, as shown here, and a phosphate, as shown here, to create a single nucleotide. The two chains of nucleotides twist in opposite directions with phosphate groups and sugars on the outside and the nitrogen bases on the inside, forming a one double-stranded helix. Phosphate, strand, twisted. There are 2 parts of a nucleic acid, a 5' end and a 3' end. This terminology refers to the carbon on the sugar. Now, RNA stands for ribonucleic acid, and its main purpose is to carry information from DNA for the synthesis of protein. It has base pairs, but instead of thymine, it has uracil, which bonds to adenine. Also, unlike DNA, it has a single helix and has ribose sugar instead of deoxyribonucleic sugar. Like a snake, it is a one-stranded helix. Protein synthesis is the process of creating proteins starting from DNA, and the flow of genetic information in the process can be understood by the central dialogue.
Central dogma,
as shown here. The central dogma was developed by a molecular biologist, Francis Crick, who also discovered the structure of DNA with his buddy John Watson. According to the central dogma, DNA makes RNA, and RNA makes proteins. Of course, the process of protein synthesis is much more complex than this. So we will get into the specifics in transcription and translation. Transcription is the first part of protein synthesis, where the messenger RNA copy of a gene is created from DNA. Messenger RNA is one of the 3 types of RNA involved in protein synthesis, and the purpose of it is to act as a messenger of genetic information for the ribosomes. However, the process of manufacturing the RNA occurs in the nucleus of the cell of eukaryotic organism because DNA is contained there and cannot leave. There are 3 stages of transcription: initiation, elongation, and termination. The first stage in transcription is initiation, which involves the binding of RNA polymers and transcription factors to a region of DNA called the promoter. To understand the promoter, one must first understand the concept of a gene. Genes are segments of DNA that contain the code for an RNA molecule.
These segments begin with a promoter and end with a terminator. The beginning part of this promoter is a nucleotide sequence called the Tata box, which helps other molecules determine where to bind for transcription. Its unique name comes from the base sequence TATAAA, Proteins called transcription factors must attach to the promoter for the enzyme RNA polymerase to recognize promoter sequences. Once RNA polymerase attaches, it untwists the DNA strands and initiates RNA synthesis at the start point at the template strand.
The next step in transcription of DNA is elongation. In this stage, RNA polymers moves down the template strand in a 5' to 3' direction, adding complementary messenger RNA nucleotides on the strand. This works the same way as DNA replication, except the complementary nucleotide for cytosine is uracil instead of thymine. A strand RNA quickly elongates as RNA polymers untwist the strands and copy the template strands as it moves. This creates the DNA-RNA hybrid complex in which one strand is DNA and the other strand is complementary RNA. When the RNA polymers hits the terminator sequence at the end of a gene, it stops adding proteins, deattaches, and the newly formed RNA sequence is released. The messenger RNA contains sequences of nucleotides that code for proteins called exons and the sequences that do not code for proteins called introns.
Because introns are unnecessary, they are removed through a process called DNA splicing. In addition to this, the guanine triphosphate cap, called a 5' cap, is added to the 5' end of the newly formed RNA, and a 3' tail composed of adenines is added to the 3' end. These modifications make it easier for mRNA to leave the nucleus and connect with other organelles later on. After these additions are complete, the RNA molecule is mature, and it leaves the nucleus of a cell through a nucleopore and enters the cytoplasm to begin translation.
Mutations are a permanent change in a cell's DNA, which leads to mistakes in translation and transcription processes in protein synthesis. In translation, a possible mutation that can occur is a point mutation, which is a change in a single base pair of DNA. A change in a single base can change a protein's whole structure, since amino acids affect the shape of a protein. This can occur when RNA polymers adds the wrong complementary nucleotides of RNA to down the incorrect nucleotides from DNA. In addition, a point mutation, called a nonsense mutation, can cause translation to terminate early, resulting in mistakes in transcription. Furthermore, a frame shift mutation can occur, which is when an extra nucleotide is added or deleted from the RNA. Thank you.
Hi, I'm Jody, and just as my friend Summer explained the process of transcription, I'm going to explain the amazing process of translation. Translation occurs in the cytoplasm of the cell because RNA must carry genetic information to the ribosomes, a structure where proteins are built. Ribosomes are made of protein and RNA, otherwise known as ribosomal RNA or rRNA. rRNA does not contribute any genetic information, but instead it has binding sites that allow incoming mRNA to act with tRNA. tRNA is a shorthand for transport RNA, and its purpose is to use mRNA to make a polypeptide chain. On the end of tRNA is an amino acid, and the other end is a sequence of 3 nitrogenous bases. There are 4 binding sites on the ribosome, 1 for mRNA and 3 for tRNA. The 3 tRNA sites on the ribosome are called A, P, and E sites. A site, otherwise known as the A site, otherwise known as the acceptor site, is the entry site for the tRNA, which holds a new amino acid to be added to the polypeptide chain. You can see the A site as the first one in the ribosome right here. The P site, called the peptide tRNA site binds the tRNA carrying the elongating polypeptide chain right here, and finally the E site, otherwise known as the exit site, for the tRNA after the amino acid is delivered. As mRNA flows through the ribosomes, as shown to the strand of RNA flowing through the large and small parts of the ribosome right here, the ribosomes read mRNA in sets of 3 nucleotides. Each of these 3 nucleotides, called codons, code for a specific amino acid. The start codon signifies the beginning of a sequence of amino acid. In almost every gene, the start codon has a nucleotide sequence of A, U, and G. The ribosome finds a tRNA with the corresponding nitrogen bases for A, U, and G for the start codon, U, A, and C. These nitrogenous bases are called anticodons, and they pair with the codons. The RNA grabs amino acid from the cytoplasm, as shown right here, which is RNA molecule, and after a pair of tRNA and RNA is formed, the RNA goes to the next binding site, the P site, and a new tRNA molecule with the amino acid attached enters into the A site. A peptide bond develops between the adjacent amino acids in the P and the A sites. And as it is formed, tRNA in the P site releases amino acids onto the tRNA in the A site and heads onto the E site without anything attached. Through repeats of this process, an elongation of amino acid occurs, such as this chain right here, which eventually creates a protein once the ribosome hits the termination sequence at the end of the mRNA. Possible mutations and transcriptions include when a nucleotide codes for the wrong nucleic acid or part of the amino acid of a protein is not coded, and a point mutation or a frame shift mutation. And that's it for today. See you later, Mr. Cummings.